Bacteriophages, also called phages, are viruses that specifically infect bacteria, and we can confirm their presence and quantify them using a tool called the plaque assay. Bacteriophages infect their susceptible hosts by first attaching to the bacterial cell wall and injecting their genetic material. Then they hijack the cell's biosynthetic machinery to replicate their DNA and produce numerous progeny phage particles, which they then release by lysing and killing the host cell. This lytic activity is the basis of a widely used phage enumerating technique, known as the plaque assay or double agar layer assay. Here, a bacteriophage mix is first prepared in a molten nutrient broth containing low-concentration agar. All bacteria used in the mix should be alive and actively dividing in the log phase of their growth, which will ensure that a large percentage of the bacteria are viable and able to form a dense lawn around the plaques. Next, this molten bacterial phage agar mix is spread over a more solid concentrated agar nutrient medium, which is already solidified on a petri dish. On incubation at room temperature, the low-concentration agar phage bacteria broth also solidifies to form a soft agar overlay. Here, the bacterial cells can derive additional nutrients from the bottom layer and should rapidly multiply to produce a confluent lawn of bacteria. However, as phage particles are also present in the soft layer, these will infect and replicate their genetic material within the bacteria, culminating in cell lysis, which releases multiple progeny. These phage progeny then infect the neighboring cells, as the semi-solid state of the bacteriophage layer restricts their movement to more distantly located host cells. This cycle of infection and lysis continues over multiple rounds, killing a large number of bacteria in a localized area. The effect of the neighboring cells being destroyed is to produce a single circular clear zone called a plaque, which can be seen by the naked eye, effectively amplifying the bacteriolytic activity of the phage and enabling their enumeration. The number of plaques on a petri dish are referred to as plaque-forming units, or PFUs, and providing the initial bacteriophage concentration was sufficiently dilute, should directly correspond to the number of infective phage particles in the original sample. This technique can also be used for characterization of plaque morphology, to aid in identification of phage types, or to isolate phage mutants. In this lab, you will learn how to perform the plaque assay for enumerating phages, using the T7 phage of E. coli as an example. First, identify a suitable medium for the culturing of the host bacterial cells and the bacteriophage. Here, lysogeny broth, or LB medium, was used to culture E. coli and the T7 phage. Next, take 3 clean glass bottles and label them with the media name, and then the first as LB broth, the second as LB bottom agar, and the third as LB top agar. Now weigh out 4 grams of pre-formulated LB powder in 3 sets and then transfer 1 set of weighed dried media into each bottle. Add 200 milliliters of water to the first bottle. Mix the contents using a magnetic stir bar. Then, using a pH meter and constant stirring, bring the final pH to 7.4 through the addition of sodium hydroxide or hydrochloric acid. Repeat the water addition and pH adjustment for the other 2 remaining bottles as well. Now weigh out 3 grams of agar powder and add it to the second bottle to make a 1.5% bottom agar. Finally, weigh 1.2 grams of agar and add it to the third bottle to make the 0.6% LB top agar. The broth condition in bottle 1 does not need an agar addition. Cap the bottles semi-tightly and then sterilize the media by autoclaving at 121 degrees Celsius for 20 minutes. Once complete, remove the media bottles from the autoclave and immediately twist the bottle caps to close them fully to prevent contamination. Keep the LB broth and LB top agar media on the bench for later use. Place the LB bottom auger to cool in a water bath that is preset to approximately 45 degrees Celsius. When the LB bottom auger reaches approximately 45 degrees Celsius, transfer it to the working bench. Next, sterilize the workspace using 70% ethanol. Next, add 450 microliters of sterile 1 molar calcium chloride to the molten bottom auger to make a final concentration of 2.25 millimolar. Gently swirl the bottle to mix. Then set out 7 clean Petri dishes. Label each dish on the bottom with the media name and preparation date. Then pour 15 milliliters of the bottom auger into each of the 7 Petri dishes. Allow the plates to set for a few hours or overnight at room temperature. Once set, the culture plates can be stored at 4 degrees Celsius for several days if needed, upside down to minimize condensation. Transfer the Petri dishes from the 4 degrees Celsius refrigerator to a 37 degrees Celsius incubator 1 hour before the assay. The day before the assay is to be performed, the E. coli should be cultured. Here, 10 microliters of E. coli culture was inoculated into 10 milliliters of LB broth. Place the bacteria to grow overnight in a shaking incubator set to 37 degrees Celsius at 160 RPM. Then, on the day of the assay, remove the bacterial culture from the incubator. Seed a fresh 10 milliliters of fresh LB broth with 0.5 milliliters of the overnight culture. Place these cells to grow into a shaking incubator set to 37 degrees Celsius at 160 RPM. Next, use a spectrophotometer to check when this culture reaches log phase growth, indicated by an optical density of 0.5 to 0.7. Once the OD reaches this level, stop the incubation by transferring the cell culture to the bench. They are now ready to be used for phage overlay assay. Phage titers can vary exponentially across different phage types and samples. So in order to count them effectively, they should be diluted to generate a wide range of phage concentrations. On the day of the assay, generate a series of phage dilutions ranging from 1/10 to 1/1,000,000th concentrations, following a 10-fold dilution technique. To obtain statistically significant and accurate data, perform the serial dilution in triplicate. Next, melt the solidified LB top auger using a microwave. Then place it in a water bath that is preset at 45 degrees Celsius for 1 hour. After 1 hour, collect the Petri dishes containing the bottom auger layer from the incubator. Label the plates with phage concentration and assay date. Then set out 7 clean test tubes. Label each test tube with the serial phage dilution number and designate one as control. When the LB top auger reaches 45 degrees Celsius, transfer it to the working bench. Now add 450 microliters of 1 molar calcium chloride to the 200 milliliter auger to make a final concentration of 2.25 millimolar. Gently swirl the bottle to mix. Next, add 35 milliliters of LB top auger and 4 milliliters of bacterial suspension to a sterile conical tube. Gently swirl to evenly distribute the cells, but avoid shaking to prevent foaming. Now, aliquot 5 milliliters of this bacteria top auger mix into each of the 7 test tubes. Then transfer 100 microliters of each of the serially diluted bacteriophage samples and control media, which should be simply media with no bacteriophage, to the respectively labeled test tubes. Swirl the mixture gently to ensure proper mixing. Gently transfer 5 milliliters of bacteriophage mix onto the respective Petri plate. Evenly spread the mix throughout the whole surface by gently swirling the Petri plate. Once all of the Petri plates are layered with the mix, allow solidification of the top layer by incubating at room temperature for 15 minutes. After completion of these steps, repeat the process for the second and then the third sets of the Petri dishes using the remaining 2 sets of phage dilutions. Seal each dish with parafilm and incubate at room temperature for 15 minutes. Place the culture plate upside down at a suitable temperature for 24 hours or until plaques develop. Here, plates were placed in a 37°C incubator for 1 day, a stimulating growth condition for E. coli and the T7 phage. Plaques will appear after 1 to 5 days of incubation, depending on the bacterial species, incubation conditions, and the choice of medium. Here, plaques were visible after 1 day of incubation at 37°C. Begin by checking the plates marked control and ensure that no plaques were formed in these plates, as this would indicate viral contamination. To determine the phage titer in the original sample, start with the plates containing the most diluted phage sample first, and count the plaques without removing the lids, marking them to indicate which ones have already been counted. Repeat the counting for each plate in every set. Some plates might have too many or too few plaques to be counted. Consider 10 to 150 as an ideal plaque count. Next, generate a table listing the plaque number values for the different dilutions and replicates. Then calculate the mean plaque number values for the dilution plates that contain the ideal number of plaque counts. In this example, these were the average number of plaques formed in the 10 to the -3 and 10 to the -4 dilution plates. Now, adjust for phage dilution factor by dividing the obtained mean plaque values by the respective phage dilution factors. Here, the average number of plaques formed in the 10 to the -3 and 10 to the -4 dilution plates were divided by their respective dilution factors to obtain the number of plaque-forming units, or PFUs, in 100 microliters of phage mixture. To convert the value to PFU per milliliter, multiply the generated values by 10, as only 100 microliters of phage dilution mix was used during the bacteriophage overlay preparation step, producing an additional dilution factor of 10. Finally, calculate the average of the values obtained from the different dilution plates. This will give the average number of PFUs per milliliter. The number of PFUs corresponds to the number of infective phage particles in the original sample.